This work generally covers what we did in the last 6 or 7 years on applying microfluidics to enable epigenomic profiling. And I will talk about what that is in a little bit. So the idea of precision medicine certainly is that you want to gather as much as you can the molecular information about the patient and then use this information to hopefully tailor the best treatment for that particular patient. And certainly this type of information in the post-genome area involves genomics and also epigenomics, transcriptomics, proteomics. So one thing I want to remind you is that the genomics is fairly constant. What that means is during, say, a treatment period of, say, half a year or 1 year for a cancer patient, the genomics pretty much stays stable during that course. And second of all, it's also no tissue specificity, meaning if you test the genomics using saliva or blood, you get the same results. And the other type of omics I'm talking about here, epigenomics, transcriptomics, they are fairly dynamic and also tissue specific. That actually provides opportunities for to monitor a disease because the disease tends to be tissue specific, and also they are fairly transient and dynamic during the disease development. So hopefully by following these kind of different layers of regulatory systems, that allows you to follow the disease itself. And a little bit about what epigenetics is. Certainly, people realize DNA sequence is very important in terms of disease development and everything else. However, people also realize from a very early point that the DNA sequence cannot be everything that dictates the development or the biology. Because if you look at the process of development, human being, from a fertilized egg, which is essentially a single cell, and keep differentiating and dividing to generate a human body, which includes a lot of cell types and tissue types with drastic different morphology for the cells and also drastic molecular biology, different molecular biology. So that means, however, all these cell types and the tissue types have the same genomics. And that means something else is playing an important role in such differentiation. And the other process you consider is things like memory or learning or addiction. For example, you listen to my talk right now, hopefully several days later, somebody asks what this talk is about, you can still tell something. That means this experience leaves a biochemical mark in your mind. Obviously, this kind of change cannot be a genomic mutation, so it has to be something that's more transient and more faster than that. So that people recognize that the epigenetics is involved in that type of process too. And if you look at the structure of chromosome, this is actually not entirely surprising, because if you look at a chromosome, you have 23 pairs of it, and it's not just a very long double-stranded DNA. It's double-stranded DNA wrapped around a protein called histone. And if you have any change to the histone, for example, attaching a methyl group to the histone tail or acetyl group to the histone tail, that affects the accessibility of the chromosome, that affects the conformation of the chromosome because it affects the change in that local area. So histone modification is one type of epigenetic mechanism that changes the transcription or the gene expression and the phenotype, but it doesn't change the DNA sequence, okay? The other type is called DNA methylation, that if you attach a methyl group to the cytosine, this is one of the 4 bases you have, 4 nucleotides you have in the DNA, then that again changes the charge, changes the conformation, changes the accessibility. So these type of changes are all called epigenetic changes, and they are involved in normal development and also disease development. It's very wide-ranging in terms of its involvement. So one important message, like I mentioned in the earlier slides, is that epigenome is very much tissue and cell type specific. That means if you analyze the established epigenome using, say, muscle cells, then that will be different from using, cell from the bones or other type of tissue. So the current effort is that people are establishing the reference epigenomes for different tissue types, different cell types, for humans certainly, and also for these important model animal like mice. And certainly the goal is that eventually you want to apply this to the clinical process, clinical world. The way you do that is obviously you want to first establish the reference epigenomes. What that means is if everything's in order, what does the epigenomic feature look like? And after that, you sort of want to establish what it looks like when the disease occurs. And then you compare the two, hopefully that gives you some so-called functional annotation of the epigenome, meaning a certain area or a certain feature is connected with certain phenotypes or certain disease state. Okay, then you can profile a patient using that patient's material. You say now which category I can put this particular patient into. That's sort of called patient stratification. You say, okay, based on that epigenome, that the patient belongs to this category. Therefore, this type of treatment will be statistically, say, by 70% effective on this particular patient. That's how you really develop this kind of tailored treatment for a particular patient using such information. Certainly, if you understand the epigenome and how that changes with the disease, that gives you biomarkers, that gives you drug targets potentially to work on for the other type of intervention you may have. And so certainly a problem you may have in that process is that in order to work with patient materials, you're limited by the amount of cells available to you. So I listed some examples. You have fine needle biopsy, several hundred to 10,000 cells, core needle biopsy, tens of thousands of cells. And, and then you may generate millions of cells if you do a resection of the tumor, or you look at the postmortem human sample, human brain sample, several million nuclei. And, but then you have the other side of the spectrum that if you try to look at circulating tumor cells, you really only get 1 to 10 such cells from 1 milliliter blood. Then you really roughly have 10 to 100 of them for any analysis you want to do. Certainly, in addition to that, again, epigenome is specific to the cell type. You probably will do some cell isolation to generate a homogeneous population of cells before you're testing. That would tend to further decrease the number of cells available to you. Finally, I think a large number of epigenomic marks needs to be tested. So each individual marker would require a separate assay. So I'll talk a little bit more about that in a little bit. So one assay we first look at is called chromatin immunoprecipitation. This is an assay for examining genome-wide histone modification. So this is a gold standard assay. The way it works is that you first have cells, you break the cells to extract the chromosome. And then because you want to be compatible with the next generation sequencing, now you need to break the chromosome into smaller pieces, anywhere from 200 to 600 BP for each piece. And now you're going to include these pieces that contain the histone, modified histone you're targeting at. For example, you're interested in H3, K4, ME3, that would be the H3 histone with lysine 4 tagged by methyl group. Then you'll have an antibody targeting that. And you'll have these magnetic beads coated with such antibody. Then they would pull down the fragments containing such modified histone. And then you can digest the histone, leaving just the DNA interacting with the histone. And then you can do either qPCR if you have a gene in mind that you want to see if this particular gene is interacting with a modified histone or not. Or you can choose to do sequencing, which gives you the genome-wide profile of all the locations in the genome has this modified histone. Okay, so the problem with very powerful assay, the problem with the assay is that it requires about 10 million cells. And like what we just mentioned in the earlier slides, you don't normally have that many cells to go on. And certainly a lot of the procedure was manual, so that requires multiple days for such an assay. And for histone modification, certainly you have different types of histone modification. The figure here shows that there are different histone tails, and at different locations, different amino acids, you can have different kind of modifications. So there are a large space to explore, really, to define the histone state, histone code also called. So there obviously have been development over the years to decrease the input of the cell number required for such assay. I think the two that are in red, that's what we developed. I think that roughly gave us, instead of using 10 million cells, we were able to work with less than 100 cells. And there was another technique, drops chip, developed after us that can work with single cell. The problem is that there wasn't a whole lot of information generated on single cell. They have about 1,000 reads per cell. So I'll do a comparison in a little bit when we talk about the data. So before we tackle this problem, one thing we thought as an engineer is obviously what would be the minimal number of cells you would require for such a chip-seq assay. Maybe there is a theoretical number you can sort of get an idea about. So one thing we consider is that you look at these library preparation kit, you need to prepare a library for sequencing. Normally it requires about 20 picogram, anywhere from 10 to 20 picogram of DNA. Okay, then you consider, you look at the epigenomic data. Okay, the data, the way you read that is the x-axis is the location in the genome. The y-axis is the signal after the chip-seq process. If you see a peak here, that means at that particular location, you have that particular modified histone at that location. So this is entire genome's profile. Then you can roughly look at, if you add all these peaks together, what's the fraction of the genome it occupies, okay? We look at that, it's roughly around 5%, okay? So if you consider that, then for a chip-seq assay, I described, you're roughly supposed to collect about 5% of the total chromatin fragments if you need to generate 20 picogram of DNA, and each cell contains about 6 picogram. The simple calculation shows if you collect them 100%, you only need 67 cells for such work. So that's what we were shooting for once we have an idea about that. So what we demonstrated in this, what we call MO-chip-seq or microfluidic oscillatory washing chip-seq, is that we were able to collect DNA better than by about 2 orders of magnitude compared to the state of art at that point. And that allowed us to look at really use just 100 cells to generate such data. And the on-chip process takes only about less than 2 hours. So the technical enabler here is very simple, as I will show. You look at the image here, we just have a single chamber here, microfluidic chamber, and we form a packed bed. So we are chemical engineers, we like packed bed. So, and then you have a single wall here that enable us to collect the DNA with very high, the chromatin fragments, I should say, with very high efficiency. Then we have a washing scheme to remove the non-specific absorption. So the process works like this. So you have a big, fairly big microfluidic chamber, and the wall here can be closed halfway, meaning that the particle will be stopped, but the water, the fluid can sieve through such a structure. So when you do that, then you can pack the microscope beads against such a structure, and you form this packed bed. And once you have that, you can flow the chromatin fragments through such a structure, and now they need to squeeze through the winding pathways between the particles, and there are very small gaps between the particles. Now this is like a sieving structure. Now you have a very high capture efficiency. By the way, the beads are coated with antibody targeting the particular type of histone in these fragments, okay? But certainly the chance is when you improve specific absorption, you target, you also increase non-specific absorption. So you have to have a very good washing to go with that. And so what we did was just mechanically move the beads back and forth in the chamber by applying pressure pulses at the two ends of the chamber. So very intuitive. It turned out to be working okay. And then after the washing, you remove the non-specific absorption. You can do that a couple of times. And in the end, you can, after washing of the beads, you can collect the beads off the microfluidic device. Then you can sort of digest the histone and peel off the DNA, prepare the library, just like everybody else. And then we can prepare a library for sequencing. And here I can show a video that shows the movement of the washing of the beads. So you can see the beads are moving back and forth in the channel like crazy. So this mechanical washing, and then we can sort of use just a qPCR before we do sequencing. It's a low-cost way to look at if that works or not. So the way we look at it is we look at enrichment, meaning we know certain locations that's supposed to have this signal, this type of histone modification at that particular gene. And then there is other area that the signal ought to be very low. And now you want to see the maximal differentiation between the two types of positive loci, what we call a negative loci. Then you can optimize the condition in terms of washing, amount of beads, and everything else. And you can see sort of under certain conditions, the contrast is better. These are good conditions. And so basically, our results show that we can pretty much collect as much DNA as we want. But if we look at the enrichment, when the enrichment good, we tend to collect anywhere from 5 to 8% of the chromatin, just what we estimated to begin with. And so we feel we are pretty much collecting it at the theoretical level. Certainly there is no guarantee that we are only collecting the good DNA or good chromatin fragments there. But we are in that neighborhood nevertheless. So this is the data. Again, the x-axis is the location of the genome. If you see a peak, that indicates at that particular location you have this type of histone modification. We look at the two types of histone modification. One is an activating mark, H3K4ME3. The other is sort of indicates where the active enhancer is, H3K27AC. So we worked our way down using 10,000 cells per assay, and all the way go down to 100 cells per assay. So you can see we did have higher noise level when you have less cells, but nevertheless, for the major peaks, we are seeing the same thing, getting the same information. Okay, I guess in that sense, H3K27AC works a little bit better, in terms of seeing exactly identical information there. Since now this is, now we can work with 100 cells, we can work with tens of cells. So that's the thing we are thinking about is, like I mentioned, there are a bunch of histone marks you want to look at. then if you really want to push this for precision medicine, you probably have a large number of patient samples to look at, different cell types and so on. So you want to work with high throughput, meaning you want to conduct these assays, multiple assays simultaneously. So one way we want to do it is that we can operate the multi-chip device in an isolated fashion. They are not communicating with each other, but we just run it simultaneously. So here, let's see. Okay, so what you can see is that we can this is so the washing step, I guess that's more pronounced. You can see they are moving together sort of in similar fashion. Then we are operating 4 assays simultaneously. If we can afford more valves, and we can easily make a bigger number of arrays working together. You can see the valves are flashing, and then you sort of remove the samples off the chips simultaneously too. So that's possible. In another piece of work, you can also start with the same sample, but then split that 4 ways, so that is a little bit tricky in the sense that if you still have packed bed, you have a lot of pressure drop. I think as chemical engineer, we can easily appreciate that. And so we changed that a little bit to have a fluidized bed, meaning that you have a flow, then you have a magnet to pull the beads, so counterbalance the 2 forces, and then you have the beads basically suspended in the solution. So that's less density, less pressure drop. It works a little bit similarly, and we can sort of make them work simultaneously, and washing and other things, other steps. Okay, yeah. To make it even simpler, what we thought is also that since we are working with, the goal is to work with a small number of cells, and then you are not generating a whole lot of chromatin fragments. Then you probably don't need a whole lot of surface area for such absorption. So we were thinking if we have a bigger channel, maybe the substrate, the surface of the channel will be sufficient for such work. So in this particular work, we call it surface chip. We just immobilize the antibody at the bottom of the glass, at the bottom of the channel, which is glass. It allows easier surface chemistry that way. We use a surface chemistry from Jim Heath's group that's supposed to attach the antibody with a high density. And then we do similar things. We flow the chromatin fragments in, and then we do a little bit washing to remove the non-specific absorption, and then we digest the histone and to get the DNA, we elute the DNA, prepare the library. Okay, the good news is obviously we found that works too, and so these are the data. I think one thing we explored is that also to use it to profile a histone modification like H3, K27ME3. This is generally regarded as harder to profile histone modification. The reason is that if you look at the feature H3, K4ME3, you largely see these individual peaks that are fairly sharp, fairly tall. But for H3, K27, you have these broad peaks. They are all connected together. This whole thing is considered a peak. And then they are fairly low too. Only when you have a very low background, then you are able to tell this is a peak. So I'm glad that we were able to do that too. I think the encoded data is what we put as a reference. This is what the data people collected using 10 million cells using the conventional approach. You can see we offer even using 100 cells, 500 cells, we offer actually better signal to noise ratio compared to their data. We also obviously compare that to the MoChip, which have the beads, have probably bigger surface area, and we see actually similar performance here. The The surface chip, obviously at this point, it basically just requires a single channel to do the assay. You have a channel, you flow the solution through, then you do these things, and then you can have the assay done. And so if you put a little bit plugging system in there, then that allows you to do multiple assays. You can also functionalize the surface with different histone marks. Then you can use the same device to look at different histone marks. So in this case, we tried a 4-channel device. We also tried an 8-channel device. You can see there is no crosstalk between these channels, and you have very good replication, among replicates, while you try to do the same assay to look at the same histone modification. Okay, so that's what we did for histone modification. So we also developed a device and technology for DNA methylation. So I want to switch gear a little bit to talk about how to look at DNA methylation using a technique called bisulfite sequencing. Okay, so the DNA methylation, this is again an established approach to do bisulfite sequencing. What that means is the kind of epigenetic change I want to tell now is that whether the cytosine is attached to a methyl group at this point. So what people found is that if you treat the DNA using the chemical bisulfite, it's actually bisulfite sodium, sodium bisulfite, then, if the cytosine is attached to a methyl group, then that group doesn't change. If the cytosine is not attached, is not methylated, then the cytosine becomes uracil. After the amplification, that whole thing becomes a T instead of a C. So basically, you're converting the methylation status into the sequence information. After that, you basically just need to do the sequencing. Then you compare that to the original reference genome. Then wherever you see the C becomes a T, you have an unmethylated cytosine at that location. So the method offers single nucleotide resolution. If you sequence deep enough, you can tell each and every nucleotide their methylation status is. Again, the problem is that you would need several micrograms of DNA. That's a lot of cells to generate such information. And one bottleneck we identified is that the bisulfite conversion step loses a lot of DNA. It destroys the DNA, and then they cannot be seen during the sequencing, and then you require a lot of DNA to start with. So before I get into the details of how we do the bisulfite conversion and the bisulfite sequencing, I want to mention a general problem for microfluidics dealing with molecular biology. That's on how you would add the new reagents from one step to the other. Because if you recall that if you're doing this molecular biology in the bench scale, you will just have your tube and then you keep adding things to it. And say for microfluidics, you normally start with one chamber. If you add things to it, that's going to flush the stuff originally in there out. So you lose your material that way. So there is one way to solve that problem that was in the earlier work of Carl Hansen's lab, that was also subsequently used in the commercial products from Fluidigm, is that you have a bunch of microfluidic chambers attached to each other. You keep them empty at the beginning of the process. Whenever you add a reagent, you open one of the chambers, then that gives you extra volume. Now you can put the chemical in there without losing the previous chemical. Certainly there are two problems. One is that if you have many steps, you need to attach many chambers. That gets more and more complex. Second is that you need to be careful about the interference among different reagents, because the reagent you use in the first step may not be compatible with the reagent in your last step. So they will be eventually mixed together. So the sort of the way they avoid such problem is that they couldn't use chemical lysis of cells. They use thermal lysis, which is incomplete, but they lived with it. And then they also need to design the chamber size so that they have a proper dilution so that the later reagent doesn't, the earlier reagent doesn't strongly interfere with the later steps. So we thought that that's a general problem if you want to treat, say, nucleic acids for multiple steps, DNA, RNA, whatever. You know, we thought maybe we want to think of something better than that. You know, so in this case, we have a fairly simple device. It looks complicated, but actually there is only that reaction chamber at the center, and it's attached to the we call loading chambers on the sides. And the connection between the reaction chamber and the loading chamber can be cut off or established by operating these valves. That's all this is. We realized that there is a gigantic difference in terms of diffusivity between nucleic acids and the smaller, reagents like ions or salt. Even you deal with a 250 BP DNA, which is not a very big DNA fragments, you're talking about 2 order of magnitude difference in the diffusivity compared to ions and small molecules. Okay, what that means is you can imagine you're doing a diffusion-based reagent exchange that the DNA will largely stay in the reaction chamber because it diffuses so slowly. But the small ions will be able to quickly get into the loading chamber, and the small ions or small molecules in the loading chamber will be able to get into the reaction chamber during the same period. So we did some modeling to prove that's the case. And so the process works like this when we do bisulfite conversion on the microfluidic chip. This is a cross-sectional view of the device. So the center one is the reaction chamber. The chambers on the sides or the loading chamber. So we would first start putting the DNA in the reaction chamber, and then we can open the valves between the reaction chamber and the loading chamber. Now the bisulfite would be able to diffuse into the reaction chamber, and the DNA would largely stay there. You suffer a small loss of 5% or something, Then after that, we do bisulfite conversion. The next step is called this desulfonation. You use NaOH. And again, you can this is another small molecule. You can diffuse that into the reaction chamber. And after it's done, the way you clean the DNA up is just by diffusion again. This time you load water in the loading chamber, then the NaOH will get out. Because while you're preparing the subsequent steps, NaOH needs to be get rid of too. And by doing all that, we found that we conserve much more DNA than the conventional protocol. That actually gave us pretty good data in terms of looking at the DNA methylation. I'm not going to go into the details here, but we, for other people using 30 nanogram of DNA, they were covering only 0.9 million of the CPG methylation features, and we can cover easily between 1.5 million of these features using 0.3 nanogram. So we're doing much better, generating much better information using much less DNA. And more importantly, we are actually covering the DNA with more coverage, more iteration of the sequencing, so that this is much higher confidence data for to work with. So the last part from my talk is to talk about some biology, and the medical insights we discovered using these technologies, because I want to really show that we can work with patient materials. We can establish epigenomes in a cell type specific manner, and we can generate real information or biological insights that's potentially interesting and useful. And so one direction I have in my lab is to look at the brain, how that functions and how the epigenomics affects the brain functions and the brain development. And because, as I mentioned earlier in the talk, that the brain activity, people have evidence to suggest epigenomics is heavily involved in there. So in this particular project, we are looking at different parts of mouse brain. We look at the prefrontal cortex, we look at cerebellum. Okay, cerebellum generally, people believe this is the location responsible for motor activities, the movement of the mice or human. And the prefrontal cortex, that's the place responsible for cognition and planning. Okay, so they have drastically different functions. We want to see whether we can see some difference in the epigenome. So we would isolate the tissue, and then we would generate the nuclei, and then we use FACS to separate neurons from glia. Glia is a supporting cell type. And then we can profile the epigenome for these two different cell types. And you can see sort of, we list all the cell types. There are, neurons from prefrontal cortex and neurons from cerebellum. It's interesting that neurons in different places of the brain present very different epigenomes. So that sort of gives you some hint about how the molecular basis supports the difference in the functions. And this also varies with different histone marks. You can see, for example, for H3K4ME3, this is a correlation, matrix. If it looks blue, that means they have a high correlation among the data sets. So you can see in that case, H3K4ME3 has a fairly good uniformity across different cell types, cells from different locations of the genome. However, it's much more differentiating if you look at the other two histone marks. So that suggests that these two histone marks are probably more closely related to the difference in the function of the location and the molecular biology. So that's one project we did. The other thing we look at also related to brain is that we want to see how the antipsychotic drug affects the brain's epigenome. Because, for example, epigenomic changes is suspected in mental disease like schizophrenia and addiction even. So sort of, but that the antipsychotic drug helps with the symptoms, so you wonder how they help. So we feed the mice, inject the mice actually, using the antipsychotic drug called clozapine, and for fairly long period of time, then we would look at the demethylation profiles from the treated mice. We have 6 treated mice. They are labeled by C. Then we have the mice injected only by the vehicle, using the vehicle. These are the controls. Then we can figure out some difference between the two groups. In terms of methylation, we found more than 500 locations that in the genome that they are different. And we found that a large involvement of genes related, have been proven to be related or involved in schizophrenia or other mental diseases. So I think there are 2 implications from this data. One is that the antipsychotic drug probably improve the symptoms by interacting with the epigenome. Second is that it's a cautionary note that when you, for example, look at the postmortem human brain samples, you need to be careful to differentiate the epigenomic changes introduced by the disease progression versus the epigenomic changes introduced by the drug, because the mental patients probably took this type of drug when they were alive. So you really need to be careful when you do such analysis, when you're trying to find a control for comparison and so on. So the other direction in my lab is on cancer epigenomics. Certainly, I mean, one project we have now ongoing is that to look at how a mutation like BRCA1 changes the epigenome. Our hypothesis is that a mutation like BRCA1 would sort of lay the ground for the sort of landscape that makes the development of cancer more likely compared to their wild type comparison. The BRCA1 mutation, so the BRCA1 is a tumor suppressor gene, DNA repair gene. If a woman has a BRCA1 mutation, then there is a 70% chance for the woman to get breast cancer, 44% of chance to get ovary cancer. So this mutation is really made famous by the preventative, surgery, by the actress Angelina Jolie, I guess in the early 2000s or several years ago. So this is a famous mutation, certainly. And the way we look at it is that we examine the pre-cancer tissues. That means sort of the patient doesn't have any cancer at that point, or maybe when they're young. So this is from the cosmetic surgery. We gather that breast tissue. And because our technology allows us to work with a small number of cells, we can divide the cells into 4 fractions. That's mature luminal cells, luminal progenitor, stromal cells, basal cells. And if you look at the histomodification data here, you can see that we sort of have the BRCA1 mutant compared to the wild type. You can see they largely look similar in terms of peaks location, number of peaks, with the exception of this highlighted area. Highlighted area, especially for luminal progenitor, you can see sort of the BRCA1 mutant looks a little bit different. And also for the mature luminal cells, you see some difference there. But certainly there is a lot more difference if you compare among the different cell types. So if you can sort of look across the rows. And then sort of, again, this is a correlation. The left is a correlation matrix we did. You can see you can roughly group, cluster the samples of the same cell type into one square there, the square surrounded by this black frame. And the area surrounded by this yellow frame, that's actually the mutant sample, BRCA1 mutant sample. You can see, from the, if you just look at the genome level correlation, it's hard to differentiate that from the wild type samples. But if you look at it more closely, those would be the data on the right. You look at each individual cell types, you can find these locations that the mutants, which indicated by the yellow line underneath the column, and the wild type, which is the black line underneath the column. And so you can see for basal cells, for example, you have 6,400 locations that you have an increased signal in H3K27AC, 1,700 location with a decreased signal. For luminal progenitor, you have several thousand of these sites too. But for the other cell types, one is the stromal cell. You immediately realize you see a much bigger number of the sites being different. In that case, you have 23,000 sites having a decreased signal just when you compare the wild type with the mutant. And then the other cell type, mature luminal, you have generally very few sites that are different between the mutant and and the wild type. So that in general suggests a sort of different involvement, different roles played by these different cell types. So we're still piecing all the other information together, to hopefully to form a complete picture to see if we can prove or disprove our hypothesis. And to summarize, I guess, hopefully I show you that it's important to do the profiling of epigenome in a cell type specific manner, but when you try to do that, it requires you to work with a small number of cells. Hopefully I convince you that microfluidics provides such a platform to do low input assays and also with high throughput, And one thing I guess I forgot to mention is that in general, certainly people talk about single cell analysis these days a lot, but in general, you can generate much higher quality data if, let's say, you work with 30 cells and use all these 30 cells to generate one profile, versus you use single cell technology to produce 30 data sets, then patch them together. The reason is obviously you need to do barcoding and all that kind of things. That loses DNA, that's information you cannot recover later, and then that decreases the data quality in general. So if the cell-to-cell variability is not critical for you, it's almost always a better route to go in terms of data quality to do what we call ultra-low input assay instead of single cell assay. With that, I will acknowledge the collaborators. I think I've been working with a number of clinicians and molecular biologists on different projects. I think certainly constantly learning things from them. They provide samples for us. I think Javier, that's a collaborator I have at VCU working on the brain, human brain and the mental disease. Ron is a collaborator on breast cancer, BRCA1 mutation. And I also work with Chang and Kaitan. These are bioinformaticians for data analysis and so on. There are other people I work with, for example, Sunitha on the CTC. We started some effort looking at CTCs. She's an expert on CTC isolation. We have some project looking at brain cancer, working with Melanie at Stanford, and we also work a little bit on organ on a chip, working with Julia at Northwestern. And I also want to acknowledge funding from different institutes of NIH. So with that, I'll conclude my talk. I'll be happy to take any questions.
Questions, especially from the students. Come on. Somebody. Okay, well, all right. How about I seed you with a question? So if you're interested in picking out phenotypes and use that as a criterion to pick out cells, do you think that's sort of doable in conjunction with your techniques? As you say,
yeah, certainly.
Sort the cells and then put in. I guess
Yeah,
the question on the last slide.
Sure.
All along about
I think so far, certainly, as I showed, that we'll be using FACS a lot. I think if that works, then that's good. I think a surface marker, that's one type of phenotype you look at. But for example, if there is other criteria, how certainly, regardless how you pick out the cells, as long as we have a sizable group, then we can do the subsequent steps of profiling the epigenome. And like I mentioned, the one thing that's relevant here is that how meaningful that isolation is for a particular biological question. Depending on how how good that isolation is for a particular biological problem, then that epigenomic profile is useful or not useful once we generate that. But the technology certainly works very well with whatever sorting step you have before that. But that tends to happen in the upstream.
I have another really general question. In your assay, how do you know what is noise? How do you assess? You have some peaks that are
Yeah,
high, low.
sure.
How do you know what's the baseline? What's the noise?
I think it's sort of the way we calibrate that is that, first of all, you have these cell types that have known database, published data.
But how do you know that's not because there are some subpopulation of cells that have a slightly different profile? And
Sure.
the reading is an average.
Yeah. I think it's sort of, first of all, we need to validate our technology first. In that case, you test a common cell line under common condition like everybody else. Then you match up that data you have with everybody else. For example, what we did for MoChip, we say, okay, we got the same data. That in terms of, you can do a correlation between your data and their data just by putting bins in the genome, sort of then that's just a cross correlation after that. So if you have a very high correlation.
You have to have the faith that the
Yeah.
control or the ground truth, whatever that you compare
Sure.
to is a
Yeah, you have to assume that.
homogeneous population, which I don't know.
Cell line, I guess, I mean, first of all, it doesn't matter it's homogeneous or not. I mean, if you have a reproducible mixture, then you're supposed to get the same profile. Okay, but cell line is as good as it gets, I guess, in terms of homogeneity. And but then once you have that data, you say, okay, my technology checks out, and then whatever other system you did, you say, okay, whatever I discover, I need to believe in that. But certainly there are certain criteria. For example, for H3, K4ME3, normally people would find, say, 20,000 peaks. I think it's a pretty wide range still, probably anywhere from 7,000 to 30,000 would be reasonable. If you're in that range, then you feel it's reasonable. If you have 700, something's wrong. If you have 5,000, something's wrong. So you have some general idea once you see the data. The other thing is certainly the reproducibility between the replicates. If you have a high reproducibility there, even for a weird sample, you say, okay, I think I can trust it.
I think when you were showing the results for the epigenome of the brain, and you had very coarse areas, and then you get these really fine cell type specific resolution, but all your cells, you have lost all the information about how they were connected to each other, or whether they were right next to each other in their particular chunk of tissue. So is there a way where you can combine or maybe extend microfluidics more upwards in the
Sure.
pipeline so that you can keep track exactly of where
Right, that's a very good question. I think certainly that gets to the part that how you do the cell sorting and the cell type isolation correctly there. I think in brains, it happens to be a very complex situation because literally you can say every neuron is different in brain because their location, because their connectivity. So yeah, if you really want to get into a very fine resolution, there is no limit to that. You sort of, I think one approach would be actually doing single cell analysis there. Basically, in that case, you gather the data from a bunch of cells. Then you say, I cannot differentiate these cells based on surface marker. There is no good surface marker either. But now I have this epigenomic data sets for each cells. I can use statistics to cluster them into groups. Then for each group, I tend to believe this is one subtype. And then you sort of you basically isolate the cell type that way. That's really after the profiling, not before the profiling. So yeah, it gets more and more complicated, certainly. But brain is certainly a very special organ, all considered. If you have a tumor tissue, maybe you're not that sensitive. The other thing is, for example, let's say you care only about tumor cells, but you have some infiltrating, immune cells in there. As long as the tumor cell is the bulk, let's say 80% of the cells you're talking about, then they are going to show, because it's a numbers game. When you do sequencing, every cell provides one copy of the genome. And I mean, if you're providing 80 copies of tumor cells, then that's going to show up much better than 20 copies of immune cells. So I think depending on the situation, you still solve every problem sort of individually. But these are certainly very relevant considerations. Yeah.
And the peak, the amplitude of the peak between different input from 100 cells to maybe like 10,000 cells.
Yeah.
It seems like the amplitude of the peak doesn't change linearly with the number that you input into your chip.
Right.
What's the reason?
A reason is that to prepare the library, you have some amplification process there. That amplification is not linear. And certainly we suspect that the bit collection, I mean, even on the chip step, it may not be entirely a linear process either, meaning that you may not extract 10 times more chip DNA if you have 10 times more cells to start with. I think our experience has been that you tend to extract a smaller percentage of the chip DNA when you have a bigger number of cells. So I think all these things contribute to so you don't have a linear process there. I think that's a relevant point is that when you compare these profiles, certainly you want to do these samples in the same fashion. Meaning if you start with 1,000 cells for tumor, then you want to compare that to the surrounding tissue, you better use 1,000 cells for the tissue, surrounding tissue too. So in the end, if you see that peak disappearing, that's a real thing you're looking at.
So how do you quantify the limit? I guess the real question, how do you quantify the limit of your
Yeah, there is no intrinsic limit. I think what I mentioned there was that to prepare a library, you need 20 picogram of DNA, right? One way to do that is you collect the DNA from different samples. Then you barcode the different samples differently, and then you pull the DNA together to give you that 20 picogram. Then you can further push the amount of each samples to an even smaller number. But certainly, you need to do that barcoding. You probably lose some DNA during that process. So there is some compromise there. But intrinsically, there is no limit. I think that the limit we see is that how low you can go and still being able to generate useful information. For example, in the case of the single-cell ChIP-seq experiment they did, if you just generate 1,000 reads, it's very hard to analyze the data. You no longer see the peaks. And it generates basically very little information. It covers a very little fraction of the genome. So that just stops being useful at that point. But I don't know if you can have a clear-cut line somewhere, what is useful, what is not useful. They did publish in Nature Biotechnology for all your care, and they were able to differentiate different cell types just based on that 1,000 reads. So I think depending on the question you ask,
All right, and
Thank you.